Hi everyone. Thanks for coming, and like Eric said, I'm going to be giving a talk on the project I did for the last 5 months that looked into invasive fish entering the estuary from freshwater sources. So for the first part of my talk, I'm just going to give a really broad general outline of invasion ecology and give some examples of invasive species in the area. Not all the examples are necessarily on Sanibel, but they're in southwest Florida or the Gulf. And then I'm going to go into some specifics on my research project, and from my results, talk about some conclusions, and hopefully we can have a discussion at the end, all of us together, about some possibilities in the future of having a more comprehensive invasive species monitoring plan in the estuary. So this slide has a lot of text, and you don't have to read all of it, but the most important thing to take out of it is just that all invasive species are non-native, but not all non-native species are necessarily invasive. In order for a species to be invasive, it has to actually compete with native species and cause either a decline in native species abundance or native species biodiversity. So what's the problem? Invasive species are such a problem because they harm the ecosystem, and they do this either through competition or predation with native species. They are one of the top threats to biodiversity worldwide. They're the top threat to biodiversity in the world's oceans, and then they're the second highest threat terrestrially, and that's just after habitat loss. They lead to this phenomenon that biologists call the global homogenization of species, or biotic homogenization, which is basically invasive species become established in many different areas, and that increases the compositional similarity between those areas. And lastly, they do cost a lot of money to remove and to spread public awareness, and because of loss of native species, that technically costs money too. So how do invasions happen? These three things need to be present in order for invasive species to be successful. They have to have a mode of transport, the characteristics of that species are really important, and the new ecosystem. So these are just some of the ways that invasive species can cross geographic barriers and become established in different places. Ever since humans have been able to travel over continents, they've been able to bring species and cross the geographic barriers that they have evolved for millions of years to stay within. So almost all invasions are anthropogenically facilitated. The first one I'm going to talk about, some intentional introductions, is biological controls. Biological controls are both the best and the worst idea that wildlife managers have ever had. There's a lot of examples of species being introduced to control a pest or another invasive, and then they end up causing more damage than the original pest would have by itself. Just like some examples that come to my mind are cane toads in Australia and the mongoose in Hawaii, which killed off almost half of Hawaii's endemic bird population. But the example I'm going to talk about right now is it's actually a more positive one. This is a grass carp, and these are considered invasive in Florida, but they've been introduced by FWC to eat vegetation in canals and waterways. Even though there's a lot of them, and they obviously get very big because they're eating a lot, they are all sterile because they're triploid, so they're not reproducing. And these days, managers look into it a lot more before they do years and years of research before they introduce a species as biological control. So aquaculture is another mode that species become dispersed. This is a tiger prawn, and this is a relatively new invasion in the Gulf. It's established in the North Gulf, but just this past year, they've had 100 reported sightings of the tiger prawn in Florida along the Gulf Coast. And people are really worried about it because not only does it have like a voracious appetite, but it carries a ton of different diseases that they're worried will spread to native invertebrates. And that's native to South Asia and East Africa. So I probably don't even need to talk about this because everyone here
it's hard to find one these days.
Has any brain in their how bad it is, but this was brought here as an ornamental plant, Brazilian pepper, and it's overtaken almost, I think they estimate it at 700,000 acres in Florida are covered by Brazilian pepper right now. And they're so successful because they grow really quickly and they have an ability to shade out other plants. This is a species that is near and dear to my heart, even though it's so terrible. But I've done 2 independent projects on lionfish before, and in a week or so I'll be starting my graduate research on lionfish. Around here, it's got a lot of press lately because they opened up the spearfishing regulations in Collier County, which have been closed forever. So originally it wasn't even supposed to enter the Gulf. They didn't estimate that it would. But about in 2010, they had their first sightings in North Florida, and now it's well established, or North Gulf, and now it's well established throughout the Gulf. And right here by Sanibel, it's offshore at reefs, but it's spread to estuaries on the Atlantic coast, so that's a good reason to think that they might do the same here. They are an amazing competitor because they eat everything. They're very aggressive and predatory, and they have an amazing tolerance, not just for temperatures, but for depth too. You can find them deeper than 1,500 feet and as shallow as 3 feet. And yeah, they don't have any predators, and wildlife managers have been trying to train grouper actually to consume them, but which is kind of helpful, but the grouper will consume them, but only if they're already dead and fed to them on a spear. So they're not actually killing them themselves, but maybe they will in the future. So the idea. And yeah, that's a picture of me. You can kind of see it if you look really hard, but spearing lionfish.
You can't eat them, or you can't eat them?
You can eat them. They're actually really good for eating. Some of the Keys population, they're worried because since they feed on such high trophic level, they have concerns about the same thing that you would have with like snapper or grouper, but I forget.
Cichlasoma.
Yeah.
Right.
Thanks.
In unison.
But they're actually really good to eat, and that's one thing that they're trying to get people to hunt them and eat them and put it on menus. So,
have you eaten one?
Yeah,
Have you eaten one? That's the question.
Have you cleaned one?
Yeah.
Huh.
The only things that are poisonous are the the anal spines, and yeah, there's 2 spines that are kind of...
What's those 2 things above his eyeballs?
Those are they're horns, but they're not poisonous or anything. There's not really any reason for them to be there. They look...
He's got iPods.
Yeah, they look very strange, and that's why they were traded so worldwide as an aquarium species, and that's how they got. They actually were established off Florida before they were established anywhere else. And yeah, now they find them down to Brazil and up to Maine in the summertime. So
Saying Goliath grouper can't eat them or can't eat them? Since we have so many Goliath grouper these days, that's ridiculous.
Yeah, I don't think they were training Goliath. They were training. I don't remember.
If they train them, why they plan on just training a whole bunch of them and then releasing them and hoping they'll
learn from example?
No, they go out diving, and the divers will spear one and feed them to them. So then the grouper actually follow them around because they want to get fed. And
now I'm scared.
They won't kill themselves. So.
Seems counterproductive.
Enough on that. Now I'm going to talk about some unintentional ways that species get here. And this is the Cuban tree frog, and it came here in the early 1900s off of cargo from ships from the Caribbean. Obviously, we have this on Sanibel. They eat everything, and they do really well in disturbed and fragmented habitats, and they've adapted to survive with humans very well.
You know what I observed when I raised frogs one summer? I was reverting to my childhood. Is that none of the other frogs would eat the tadpoles of that one because it has so many chemicals in it that the other frogs could not.
Poisonous mucus or something. So they're not really good pets for people. They're not frogs. And this last example is a way that humans are indirectly affecting invasions, and this is something relatively new that I actually learned about when I went to a conference with Rick and Mark at a benthic ecology meeting. And this is a black mangrove, and normally they're limited by their temperature tolerance, so with climate change they've been able to expand their range, and they're overtaking salt marsh habitat in the North Gulf, and this is something that they're just starting to publish research on, and everyone's kind of up in arms about it because there's a lot of species that are they're specifically evolved to survive in salt marsh, and they couldn't in mangroves. So now I'm going to talk about some characteristics that almost every invasive species has. The first one is tolerance, and that's really broad, but it can be applied to abiotic and biotic factors. So they have tolerance for temperature, depth or altitude. They can go a long time without eating oftentimes. They also have a generalist diet, which means they're not specialized. So they can, if one thing's not available, they can eat something else. And usually they have some type of aggressive behavior, or they're predatory. They're pretty much bullies. And there are strategists, which is an ecological term that means they don't invest a lot of energy in their young. Instead of having parental care and only one young, instead they just have as many as they can, and they don't really have a season. They can just keep reproducing. So, yeah, that's usually not so great. And all these things together make invasive species really good competitors. So, some ecosystems are more vulnerable to invasions. Florida in general has the most introduced species in the continental U.S., and there's a couple reasons for that. The top one is its temperate climate. It's warm here, especially in South Florida all year long, and so cold tolerance isn't really an issue for species, especially when we're coming from South Asia or the Caribbean or Central America. They're used to being in warm weather, and we have that here. Another thing about Florida is that we have very lax exotic species ownership laws for pets. So, like the Burmese python and Nile monitor lizard and all those were originally pets, exotic pets, before they got introduced. And it doesn't look like Florida is going to change that very soon. And also, we get so many travelers and visitors here compared to any other state, so that's more opportunities for people to bring in species here. And then the next thing I want to talk about is islands. From a biogeographical standpoint, islands have simple trophic levels and simple food webs, and their species have evolved to have poor competitive ability and anti-predatory adaptations. So this makes them especially susceptible to invasions. Now, Sanibel isn't... The thing about Sanibel is it's an island, but it's not so far from the mainland that it's completely isolated, so they do have some mix occasionally, but still we do have a really subtropical climate here, and we're home to a lot of endangered species, so that's definitely a concern when you're talking about invasives. This is just a basic timeline on the introduction of species and then when their population booms, and it looks at eradication and public awareness as factors. So pretty much usually after a species is introduced, it lays low for a while and no one really knows about it. It will get detected, but it won't really either be brought to the rest of the public's awareness or even like the government's awareness. So once they hit that middle stage, they just boom and they grow really, really fast. And by that time, people notice it, but it's really hard to eradicate it. So that makes it really difficult. And even though it looks or sounds impossible, there have been some successful eradications, especially in isolated areas like islands. But every eradication has certain things in common. Money, obviously. Biologists that know a lot about the species, either their ecology, their biology, their range. The public has to have a part in it. They have to be willing to report sightings and whatever, or else it's going to make it way harder because managers can't just do that on their own. And they have to have a controlled reintroduction pathway, because if they're just going to get reintroduced, then it's a whole waste of time to even go through the process of trying to eradicate it. And obviously, with early detection, you have a higher chance of eradication. And that's just a map that I thought was cool because it looked at the main pathways, how invasive species, or marine invasives, transport themselves and are introduced, and it also has areas that have really high amount of invasive species, and the Mediterranean looks like they're winning in that. But now I'll talk about my personal project here and go into some specifics. We decided to do this project in particular for several reasons. First of all, I'm very interested in invasion ecology. Southwest Florida and Sanibel have an invasive species problem, and I know we've been doing a lot of research in the dunes lakes for quite a while, and we wanted to get a better idea. We knew there was invasives, but we wanted to get some population or species composition estimates, and we knew there was some weird things going on in the dunes. So the dunes are a stormwater lake system, and they can't even really be considered lakes because they're not completely freshwater. Here I am showing, I don't know if I have a pointer, but this top one is Lake 4. This is where we did a lot of our sampling, and it's connected to the estuary right here, and all the lakes are also connected to each other. So in really high tide events, water will come in from the estuary over that weir, filling Lake 4 and then the culvert into the other lakes, and every lake in there is saline at some level, but Lake 4 is just the most saline. And also in rain events, water floods and gets pushed back over the weir into the estuary. So this offers a way for invasives in the lakes to get into the estuary. And another thing I want to highlight is that this is just a case study. This is happening all over South Florida. There's invasives in rivers, lakes, all types of things. There's tons of ways for them to get into the estuary. And this paper that I mentioned here that I have with me, if anyone cares to look at it, it's a study done 2 years ago by FWC folks at Charlotte Harbor, and they found a significant amount of introduced freshwater species in the estuary. And they were looking up in Charlotte Harbor. They looked in the Caloosahatchee. They didn't get down as far as Tarpon Bay, but there's definitely species there, so it's worth looking into. So these are my two focused species. This is the Mayan cichlid on the left and blue tilapia on the right. Before I talk about their biology, I'll just mention some taxonomy. They're from the class Actinopterygi, which is almost all fish belong to. They're from the order Perciformes, which is perch-like fish. Their suborder is Labridae, which in this suborder there's cichlids and then wrasses and parrotfish. So if you're familiar with those species, they do kind of look similar, especially in the shape of their mouths and heads. And then they're both from the family Cichlidae, which is one of the most diversified families. And there's over 1,500 species that belong to that family. The reason why they're so highly diversified is their pharyngeal jaw, which fish have actually two jaws. They have an oral jaw on the outside and a pharyngeal jaw on the inside, and usually they're fused, but in cichlids they're detached. So this allowed their oral jaw to be able to specialize on what food they want to eat. Think like Darwin and the finches, how their mouths were specialized to what they wanted to eat. So, and something else that characterizes that family is that they're extremely territorial. So, these are just, I won't go over all these things, but these are some comments on their biology. Both of them consume phytoplankton, algae, detritus, plant matter. They're not thought of as predators in the sense of eating other fish, but they will eat fry and small eggs and invertebrates. So this tilapia is native to North Africa, and it's been introduced all over the world because of aquaculture. And this is just one species, the blue tilapia, but there's, I think, over 20 species that have been introduced worldwide that are tilapia just from aquaculture. The reason why there's not a whole lot of research done on blue tilapia is because most other places in the United States, Mozambique tilapia and Nile tilapia are the invaders that people are most concerned about, and blue tilapia is it's a really big deal in Florida, but that's about it. Just about their reproduction, they are mature really early, and then they're mouth brooders. So they make a nest, the female releases the eggs, the male fertilizes them, and then she takes them into her mouth to protect them. And even when they're hatched, the fry will still come back into her mouth when they perceive a threat. And they're considered the most widespread introduced species in Florida. That's just by area, but not necessarily by abundance. And then the Mayan cichlid is native to Central America, and they don't really know how it was introduced, but they think it could have been by aquarium owners because cichlids are pretty popular aquarium species. They were first found in the Everglades in 1983, and then most of the research since then has focused on their range expansion. So they've been expanding their range northward and outward from the Everglades since then. And then that paper I was talking about before has found them in tidal areas in the Charlotte Harbor, and I think in Tampa Bay they've also been found. And they are not mouth brooders, they're just substrate spawners, and the parents will guard the nest for a small amount of time after being fertilized. And like I said, there's a lot of knowledge gaps. I talked about how there's not a lot of information on blue tilapia. There's also the only information on Mayan cichlids is the range expansion, and after that, there hasn't been anything, testing their salinity tolerance or their likelihood for establishment in saltwater estuaries or whatever. So it's something interesting to look into. So what I did for my project is I had a field portion and a lab portion. The field portion, all we were looking for was absence of invasives. We wanted to see if there was any in the estuary that we could find. And we also wanted to get species composition of the dunes lakes. So even though, and then Ladyfinger Lake was on the map before, they're not actually lakes, they're just part of Tarpon Bay. So those were the two areas that we did our staining sampling. So I also did a lab project, but I'm not going to talk about that until I start talking about the methods because it'll just get confusing. Okay, so for our field research, we pulled this large stain. It's 21.3 meters long, 1.8 meters wide, and it has a 3 millimeter mesh size. So anything smaller than 3 millimeters can pass through it, but anything bigger than that is going to get caught in it. We did 35 stains altogether, either in the lakes or in the estuary, and I think this is a map of each place where we pulled the stain at. And so here's the weir again, and there's also, this is Dixie Beach Road. There's culverts in Dixie Beach Road that allow this water from this area, which I think is Sandoval property, to flow into Ladyfinger Lake. So we kind of tried to look a lot along that area to see if we could find anything. But unfortunately, we didn't. We did find a lot of cool stuff, like over 24 different species, and these are just some of them. That's a juvenile redfish. And this is a snook that actually came out of the dunes lakes.
And tarpon, then, huh? And tarpon?
We did catch tarpon. I just didn't put the picture in here. We caught it casting, not staining, so yeah, but it's pretty crazy that they have so many marine fish in that lake. They have blue crabs, tarpon, snook. I mean, even the small fish, gobies and silver sides and whatever, those are all marine species, and fish aren't really there. And this was another thing. In Ladyfinger Lakes, they had an extreme macroalgae bloom. So that was just an observation, and I think Eric has been talking about maybe looking into that and doing a project on that in the future. Yes. So here's some of our results. If you want to understand this graph, you can ask Mark.
Really?
No, I'm just kidding. It's a multivariate analysis, and basically what it's testing is similarity. So these are our lake samples down here, and they're all very similar to each other as far as the amount of species that are being caught, the types of species, and then the estuary samples are a lot more variable, which kind of makes sense because you're getting all different types of species. So we wanted to compare the estuary and the lake, the dunes lakes, a couple of different factors. This is just the mean number of species that you get per stain, so that was significantly higher in the dunes lakes. And also the mean abundance per stain was higher in the dunes lakes, so those are both significant. But the important thing to note here is even though this is like 360, we're getting a lot of little fish. We're not getting 360 like snook or tarpon or whatever. So the lakes themselves are very productive and they can support a really large population, especially in those small little grazer fish. And this is the last thing we kind of looked at, which is diversity index, which surprisingly the lake also had a higher diversity index, and that was significant as well. So all these things indicate that, like I said, the dunes lakes are really productive, and that you have a lot of similarity in the sands that you pull in the dunes, but there's nothing really to expect in the estuary. And obviously we only did 35 sands, so that's kind of a limited data collection. So we know from anecdotal information that there's definitely invasives out there, but we just didn't catch them from our sands. So because we still know that they're out there, we did a lab portion, and both the species I previously talked about, we subjected to different salinity treatments. So the first treatment was a control, and that's at 10 parts per thousand, which for freshwater species sounds high, but they're coming out of the lakes where it was 10 parts per thousand, so we wanted to keep them as similar to the environment they were coming out of as possible. And then the other two treatments I subjected them to, one was a wet season estuary estimate that we had from data that we were taking around the Ladyfinger Lakes region, and then this one was a dry season estuary estimate. And for each treatment, we subjected the fish to higher salinity and observed them for 2 weeks. So we collected our fish 2 different ways. Most of them were caught by cast net, but some of them we caught with the same seine that I showed before. This is the fish shed that we have out back in the marine lab, and this was our experimental area. There's 3 tanks on the left and 3 tanks on the right. They're separate from each other. They're maintained on one filtration system. They're each maintained on separate filtration systems. What we did is we put 6 fish into each tank so we could have replicates. And when we brought the fish into the shed, they were drip acclimated to tank conditions. Even though it was a similar salinity, there's still other things that could be different. So we drip acclimated them for about 2 hours. And like I said, for the first couple days, they were always given a couple days to kind of get used to the tank conditions, and they were maintained at 10 parts per thousand. So before we started any trials, we caught them, weighed them, measured them, and fin clipped them for identification. So we actually have a lot of data on each individual fish's growth and like when each fish had died. So it's pretty helpful, even though we haven't had time to look into that incredibly yet, because this just finished a couple days ago, actually, my last trial. But one important thing that I wanted to note is that we wanted a similar size distribution for every treatment. So we caught 18 separate fish for every treatment that we did, and you don't want to have like a bunch of small fish in one, a bunch of larger fish for the next one, because you want to be able to equate it as best as you can. So in general, this is bin size for total length. Most of the fish were around these sizes. They all look pretty similar, so that was good. And this is the tilapia. And then the cichlids, we actually had a lot more smaller fish, but like I said, we tried to keep it the same every trial, that we had the same amount of smaller fish and larger fish that we were subjecting to different treatments. So at the beginning of a trial, we would raise the salinity, and we raised it pretty much as fast as we could, and I have a video. Yeah. So this is the apparatus that we used to raise the salinity, and we built that pretty much so that we could raise the salinity into the 3 tanks at the same rate. And a couple other things about the tanks that you can probably see. This is the cichlid side. I put these buckets in so that they would have covers, because they're very skittish and it would make them less stressed. I wanted to eliminate as much stress as I could. Also, I fed each of the fish, each side of the fish bed every day. The cichlids got plate food, and the tilapia got a mixture of plate food and culture food, but they like the plate food better. And this water is actually from a tidal creek right outside the marine lab, and we pumped it into these tanks, and then we pumped it through that filter that you just saw, which had a 100 nanometer and 20 nanometer filter size, just to make sure we could get out anything if there was like red tide in there or something, that it would be able to filter it out and that wouldn't affect the fish. So this is just from the short time that we increased the salinity. To raise it to 23 parts per thousand, it took approximately an hour and a half each time. They took a data point every 30 seconds. So, and we would, after we put in 200 gallons, we would let the tanks kind of mix. So we would turn off the hose that was filling up the tanks with the salt water. And then to raise it to 35 parts per thousand, it took about 4 hours. And sorry that it has a left and right on here, but the left is tilapia and the right is, no, the right is tilapia and the left is cichlids. But they're very similar anyway, so it doesn't matter. So during the trial, I checked the fish tanks once every 12 hours. At the beginning, I checked it more often than that, but I kind of, and I also checked the water quality periodically, like every 3 days. And this is just the average water qualities and then the standard deviations, because I just wanted to show that they didn't change that much. We didn't have like a huge spike in temperature or a huge loss in DO that could have caused them to die. So we wanted to make sure that their reactions were due to the salinity treatments only. And this is a behavior that I observed quite frequently. And what it is they started losing their equilibrium because they couldn't really adapt to the salinity. But they weren't dead. They were just laying on the bottom. But they were, like on their way to dying. And a couple hours later they'd be dead. But it was something definitely interesting that that's the behavior they exhibit. So these are my results from the salinity treatments. This is a tilapia, and as you can see, they all died at the seawater temperature. It took them about 2 days, and they all died. And then this is the estuary one, which we actually had about 50% survival in the wet season estuary, that is, which is 23 parts per thousand. And then even in the control, we had some deaths. And there's a bunch of different reasons. And I think the fish were already stressed coming out of those lakes. It's just not like the ideal environment. They had some parasites, leeches that were on them, so that could have caused maybe some mortality. But when we did the stats on them, this is just looking at mortality instead of survival, so that's why 35 parts per thousand is the highest. We found that the 35 was significantly different from the control or the 23. This is pretty important. It kind of shows that the tilapia can survive in the estuary during the wet season, and they even might be able to find freshwater refuges during when the salinity gets really high. But most likely, they wouldn't be able to establish and reproduce and survive there year-round. And I was going to do some graphs on the cichlids, but it's kind of pointless because this orange line is the survival for every treatment for the cichlids. I didn't have one cichlid die the entire time for any reason, even like if they got smashed bringing them back to the lab or something, which they didn't, but they just I didn't have one cichlid die the whole time. So since we couldn't really analyze anything with their survival, we did have time to look at their growth. And this is kind of interesting because their mean growth was positive with every treatment. So even at that, the 35 parts per thousand, they were still growing. Even at the 23, for some reason that one's a lot smaller than the others. And you can also see the variance is very high. There's a lot of fish that are either growing a lot or growing a little, and it's really variable. But yeah, altogether, they didn't really have a problem with any of the salinities, and they were still growing, so that's some results that are really interesting. And altogether, the cichlids definitely will have a higher risk of establishing. There's anecdotal information, like I said, that they were already out there, so it's definitely worth looking into to see if they continue to populate in the estuary. I think right now, even though they can survive in the salinity, they still prefer fresh water, so they might stay closer to Caloosahatchee or something. But once they become really abundant, they could get out of that area because they don't want to compete with all their brothers and sisters and whatever. So they will have a higher risk of spending the energy to acclimate to the higher salinities to avoid that type of competition. And the tilapia have a smaller risk of establishing, but they can still survive pretty well in wet season estuary salinities. And there's a lot of places, there's a lot more things that I wish I could do, and more research that's needed into it. The cichlids, even though we couldn't look at their reproduction because not all of them are reproductively mature, most of them weren't actually. So Mayan cichlid reproduction at higher salinities, we don't really have any data from. And no studies that I've seen have really looked into their reproduction. Some of them suggest that they're not reproducing in the estuary yet because there's only small fish and they think that they're just getting flushed out from freshwater sources. But there's not enough information to make any type of firm confirmation, so that needs to be looked into. Like I said, the possibility of them staying in freshwater, even if they can survive in the salinity. And what I hope to get out of this was for people to see that it is a problem, and I don't know the details on the refuges monitoring in the estuary for species right now, but I think it should be something that they do frequently because not only these two fish, but this paper found like 8 different freshwater species in Charlotte Harbor, and then we have lionfish, and there's a ton of other invasives, and it's just something that they should look into, and you guys can let me know if you think that they should look into it too. And yeah, that's why thanks to SCCF and all the donors, and I wanted to acknowledge Eric for helping with my experimental design. Jeff helped me catch and measure fish. AJ helped me run the salinity treatments in the tanks. And Rick taught me how to cast net and cast that with me. And Mark was probably the most involved person in my project. He was out in the field every single day, and he helped me a lot with data analysis too. So thanks to all of them, and I'll take questions.
How many cichlids have you guys caught in the last, say, year in the San Isabel River?
Well, we don't do the river. The place I've been catching them. Well, we started finding them in Haas Pond, which is connected to the river. I guess it's all connected. I think it was 2011, the fall of 2011, when I first caught cichlids in there, and then the place we catch them over on Johnston's track. So we catch. We do our sampling during the wet season, so it's from, it's actually going to start probably next week or in 2 weeks, start doing that. And we catch them on Johnston track. We catch, probably about, on the east end we catch more abundant. We usually catch throughout the season a dozen or so of them, but they're, the little babies.
So if they ended up out in the estuary, what would they harm?
Well, since they're not predators to larger fish, mostly it's just going to be competition-wise because they can eat all different types of things. They obviously can reproduce in crazy amounts because we find them in the lakes in crazy amounts. So it's really competition for space and resources that they're going to affect as far as native fish go. And even though they're small, they can, in large numbers, could still be a huge problem.
Yeah, like what she said, in freshwater systems, they pretty much displace bass and sunfish. Bass for sure, but, and there's papers. People have studied that, and they at every stage in their development, they outcompete those two fish, those two, family, or that one family. Okay, those fish. So they're really good at just, and they gulp air in low oxygen conditions. They're not as susceptible to low DO and stuff, so they can survive through things that a lot of other native fish probably can't survive through. So, and also they in freshwater systems, they target zooplankton a lot, and that can screw up what's going on and, you know, the ecology of the system, and also who knows what could happen out in the estuary.
So do you think they go here on their own then, rather than, you know, if they could survive the cichlids?
What do you mean, from the Everglades or just?
Yeah, from like Caloosahatchee or something like that.
Yeah, well.
Because Chris seems to think that somebody released them out here, but you know the cichlids.
I don't think so.
Yeah, the cichlids weren't, because especially too because what he was finding is that like for years when he was doing the fish sampling, we weren't finding them west of Tarpon Bay Road,
Yeah.
and then recently we did, so it would make sense. And I don't think there's no access for the freshwater. There's no access on the west end, right, to the Gulf. It's all on the east. The west of Tarpon Bay Road, right? It's all the freshwater is the weirs on the east end. So
The Tarpon Bay Weir is west of Tarpon Bay Road.
And there's the I mean, does that thing ever get breached by a high tide?
I
Yeah,
remember.
they can move from the east end to the
Yeah,
west end
definitely.
on certain.
And that's happened, that water exchanges happen more frequently.
Yeah, but I'm saying, so there's no, like-
From a
like the
management
weir that
standpoint.
Right.
Yeah, not at like Wolford or anything. They wouldn't come in.
yeah.
So would you recommend raising the level of the weir, putting a fence over the weir so fish can't get over, or?
I don't think the weir is really a concern because they're going to be entering from a bunch of different places. I think more research into if they will actually reproduce and stay in the estuary all year long, because I think it's definitely a possibility, especially if their numbers get so high that they want to avoid, crowding and competition with other cichlids. But I don't think that's going to solve anything to just focus on the weir.
I think there's a paper or something out there about how Mayan cichlids have destroyed habitat in the Everglades.
Oh,
Yeah.
So
that's what
I
I
mean,
was
that's-
going to ask. If
but,
there
you know,
was any
that's
other studies.
different than
Yeah, because they
Yeah.
root around in the substrate, right? So-
yeah. I mean, they cause a lot of different kind of problems. Yeah.
Yeah, they're both, like I said, they build nests, and that's mainly what they're talking about in that paper, is that their building of nests is uprooting things.
But luckily, they're really, really tasty.
We just need more of you
What,
to
the
eat them.
cichlids or the
Both of them.
Both.
Both of them. They pretty much taste the same.
One
And they're
question-
easy to catch, and
Why couldn't you catch them in your seine?
We caught them in the lakes, but our seining in the estuary was more difficult. Just pulling a seine there is harder because you don't have really shallow surface all the time to close it and pull it
They
in.
go insane, too.
And it's easy for them to escape because, like I said, there's no shallow area to kind of.
they
try
go
to
back
control
up in the
them.
mangroves or whatever. We can't get
Yeah.
up in there. And it might have been the wrong time of the season if we really,
Yeah.
I mean,
Wet
probably
season
wet
was-
season is when there might be more exchange with. So.
And unfortunately, I wasn't here during wet season, but that's something that is definitely would definitely be better than looking now or in the dry season, because you have more overtopping, you have more fresh water for them to survive in. So that's the thing.
I mean, you can really see from the
Someone will
species
think-
that are in that lake
Not me,
that
but
there's a
someone.
lot of exchange both ways going on there, and those lakes can all, I mean, that lower lake, the one that we did most work in, I mean, it's 10 parts per thousand salinity, so it's almost like an estuarine environment rather than a freshwater lake environment. But then as you go up in that system, the lakes get fresher and fresher as you go upstream. So they kind of have, the fish in the dunes, for example, I mean, just what we seem to know is if you go upstream in those lakes, you catch some tilapia in the more freshwater ones, they look really healthy, and there's just anecdotal information, there's fewer tilapia fish kills up there, but then in that lower lake where we were seining, 10 parts per thousand, we saw lots of evidence of tilapia fish kills and all, whereas we didn't see it in the upper lake. So what, and the dissolved oxygen is probably higher in that lower lake. So they're under more, to us it just looks like they're under more stress in that lower lake, being in higher salinity waters, the tilapia at least.
Yeah, I don't know if you can see it in this picture, but what we would see a lot is that they would get lesions. Yeah, you can see it in that picture. After being subjected to the salinity for a certain amount of time. So once they were dead, they had like lesions all over them, I think, as a result of stress. Yeah, the ones coming out of the dunes lakes definitely weren't healthy. They were really skinny, or they were we saw a lot of deformed ones, which we didn't take, but they would have like giant growths, so their mouths would be all scarred and stuff.
But there's chemicals that go in those lakes probably too.
Oh, I'm sure there are lots of them.
Well, thank you all for coming. Stick around if you want to have pizza. Probably not here yet, so.